Hi, thank you. To begin, I would like to ask who here had coronavirus or knows somebody who had coronavirus. You probably don't need to look around to know that almost everyone, if not everyone in this room, has raised their hands. In 2020, with the outbreak of SARS-CoV-2, the virus behind the COVID-19 pandemic, the world was reminded of the public health dangers posed by novel virus outbreaks. COVID also highlighted the importance of understanding the way different viruses attack humans and propagate. But what is a virus? A virus is an infective microbe that's made up of genetic material, either DNA or RNA, surrounded by a protein coat. Viruses are the most numerous biological entity, and there are many, many different types of viruses which infect all forms of life. Viruses, although they're so prevalent and so good at reproducing, they're not able to do so on their own. And in order to reproduce, they need a host, the cells of the organism which they infect. The goal of a virus is to reproduce, to make new viruses. But as we just said, they can't do it on their own, and they need a host cell. They come to the host cell, they take over its mechanisms, and use that to create new viruses, which go on to infect more hosts and spread the virus. And in order to understand how the virus uses the cell mechanisms and takes over the cell, we first need to zoom into the cell without the virus and understand what's going on. Our cells are responsible for carrying out all the functions of life, and they do so through molecules called proteins. In order to create the right protein at the right time for whatever it is that the cell is trying to do, the cell needs a set of instructions, and that's contained in the DNA of the cell. And that set of instructions then goes through a number of steps before it's made into the desired protein. In order to better understand this process and all these steps, let's compare our cell to a bakery. Our bakery at any given point in time needs to make a certain protein, some sort of cupcake. The recipe for that cupcake is contained in the recipe book, in the DNA of the cell, and that recipe book contains all the recipes for everything that the bakery makes. And because it's such an important piece of information, that recipe book is kept in a separate room called the nucleus. When the cell gets ready to make the protein that it needs, it takes the set of instruction and copies it through a process called transcription into an mRNA molecule. This mRNA recipe is then exported from the nucleus into the rest of the cell, where it meets the ribosome. The ribosome is the chef of the cell. It reads the mRNA recipe, gathers and mixes all of the different ingredients, and it bakes the final protein through a process called translation. Just to simplify the analogy, our cells take these mRNA recipes, the ribosome reads the mRNA, and it bakes the final protein cupcake. When a virus infects a cell, it does two main things. First, it stops this process. It prevents the cell from reading the recipes and making its own proteins. And this process of stopping the cell from making its own proteins is termed host shutoff. The next thing the virus does is it introduces its own viral recipes and forces the cell to make new viral proteins and new viruses. The purpose, from the virus's perspective, host shutoff and introducing viral recipes serves two purposes. First, it takes all of the resources and mechanisms of the cell, and instead of having to share those resources with the cell, it takes them all for itself and uses all the resources to make viral proteins and new viruses. The other thing it does is by shutting off the cell's ability to create its own proteins, it prevents the cell from mounting an effective antiviral response. Previous work in our lab showed that when you infect a cell with SARS-CoV-2, the virus achieves host shutoff through a few different mechanisms. First, it prevents the mRNA recipe from leaving the nucleus and entering the cytosol, entering that same kitchen of the cell. It also binds to the ribosome and physically prevents the ribosome from reading the recipes, preventing the ribosomes from baking those final proteins. And finally, the virus causes the mRNA recipes to be physically cut. Our current research focuses on this third mechanism. And we also showed in research we did in the lab that it's sufficient to achieve this cleavage to have one viral protein called NSP1. And while we know that NSP1 causes this to happen, that it causes the mRNA to be cut, we don't know who is responsible for the cutting or the mechanism behind the cutting, which is the focus of our research now. We want to understand better who the molecular scissors who come and actually do the cutting are. And also, we want to understand where the cutting is occurring. We want to know where on the mRNA molecules and different features of the cleavage in order to get a better understanding of the mechanism behind it. To answer the first question of who is involved, we use CRISPR-Cas technology, which is, as was mentioned earlier, it comes and it perturbs a specific gene. We choose which genes we want to perturb. It perturbs the gene, and then we can look at what happens in the cell in the absence of that gene. What this means is that we're taking a page from the recipe book and we're ripping it out. And that means that whatever protein was supposed to be made by that recipe is no longer made. And now we can look at the cell and see what happens. And by inferring how the cell responds now without that protein, we can understand about the role of that protein. In our case, we use a fluorescent reporter to track changes in the cell. And this allows us to easily read the output of our experiment. Normally, when we introduce NSP1 to a cell, we get cleavage of the mRNA, and we don't see any fluorescence. When we introduce CRISPR-Cas against specific genes, we can ask whether they're involved in this process of cleavage. And one of two things can happen. Either the gene that we're targeting is not involved, in which case we introduce NSP1, and we get the same thing. The mRNA is cut, and we don't get any fluorescence. On the other hand, if the gene that we're perturbing is critical for the process of cleavage, then when we introduce NSP1, it won't be able to cleave, and we'll see fluorescence. So if the first question was asking who is involved in the cutting, the next thing we want to ask is where are the mRNA being cut, and what are the features of this cutting? So we're trying to ask if there's maybe a subset of mRNA molecules being cut, and if so, what is common between them? And also, we're trying to ask where on the mRNA is the cleavage happening? Is it maybe only in the beginning of the mRNA, only at certain letter combinations? And in order to answer this, we use a sequencing technique called pair-seq, where we're able to take all the scraps of cut-up mRNA in the cell, and we sequence them to allow us to read the beginning of each of these cut-up molecules, and we can then map out where the cutting happened. And by aggregating that, we can create a picture of where the cleavage is happening and to infer things about how it's happening as well. Just to quickly summarize, we said that viruses rely on the host cell mechanisms to reproduce. We said that in order to effectively reproduce, the virus must first shut off and supplant the host cell's protein production through host shutoff. We showed that SARS-CoV-2 performs host shutoff through 3 different mechanisms, one of which is that it cuts up the host mRNA. And this is really interesting academically, but why do we care? And the reason why it's important to understand the nitty-gritty details of specific mechanisms like the mRNA cleavage is that understanding it can allow us to design and develop specifically targeted treatments that can help us treat not only COVID, but also future novel coronavirus outbreaks that utilize similar mechanisms. Thank you. If anybody has any questions, I would love to answer them.